Biologics and Biosimilars, Extrapolation of Indications, from the Pan-Canadian Oncology Biosimilars Initiative. This presentation has been prepared by Cancer Care Ontario and is part of the Pan-Canadian Oncology Biosimilars Initiative, funded by the Pan-Canadian Pharmaceutical Alliance. This presentation aims to educate clinicians, oncologists, and pharmacists on the topic of biologic and biosimilar medications and how extrapolations of indications can be justified. The learning objectives are to define biologic medications or biologics, biosimilar medications or biosimilars, and extrapolation from Health Canada's perspective. It is also to understand the history of extrapolation in the European Union and review a case where extrapolation has occurred with a biologic medication. This presentation will state Health Canada's guidance for clinical trials as well as review the rationale of the selected endpoints for biosimilars. What are biologics? A biologic is a complex protein molecule created inside a living cell or system. This living cell or system can be a human or mammalian body, a bacterial cell, or a cell line from a mammal. Examples of this are transgenic rabbits, E. coli bacteria, or Chinese hamster ovary cell lines. These living cells or systems can produce the exact same protein, in other words, the exact amino acid sequence and higher level structures. Biologics help to treat diseases and medical conditions including cancers, arthritis, psoriasis, inflammatory digestive disorders, growth disorders, and diabetes. What are biosimilars? A biosimilar is a biologic drug that is highly similar to a biologic drug that was already authorized for sale, also known as the reference biologic. There are no expected clinically meaningful differences in efficacy and safety between a biosimilar and its reference biologic. A biosimilar may enter the market after the expiry of the patent and data protection of its reference biologic. Biosimilars are not necessarily identical to its reference biologic, but based on Health Canada's guidelines and approval standards for the pharmacokinetics, pharmacodynamics, safety, and clinical efficacy of biologics, the two are highly similar. What is extrapolation per Health Canada? Extrapolation was previously used to refer to the authorization of a biosimilar for indications where clinical studies were not done. Health Canada now uses the term authorization of indications when referring to a scenario where a biosimilar sponsor may request authorization for all indications held by the biologic drug authorized in Canada to which a reference is made. A biosimilar sponsor is not required to apply for all indications that the reference biologic holds. The reference biologic may have patents that expire at different times and may not wish to apply for an indication for that reason. A biosimilar sponsor may also reach an agreement with the reference biologic manufacturer on the existing patents in order to apply for an indication. The decision to authorize the requested indications is dependent on the demonstration of similarity between the biosimilar and reference biologic drug based on data derived from comparative structural, functional, non-clinical, and clinical studies. In oncology, extrapolation has been used in the EU for more than 10 years. For example, the biosimilar filgrastim Zarzio was approved based on a study in patients with breast cancer who are at high risk of developing chemotherapy-induced neutropenia. The other indications of the reference biologic Neupogen were extrapolated to the biosimilar by the European Medicines Agency. Since its approval, there have been other studies conducted post-authorization with Zarzio in the extrapolated indications demonstrating the safety and efficacy of the biosimilar. The following is a case study on extrapolation. In this case, a biologic manufacturer makes a significant process change in the production of its biologic. In the early 2000s, a company who produced an innovative biologic drug wanted to change the manufacturing process to increase efficiency. This biologic drug had been in clinical use for cancer and non-cancer indications without any issues. This company was required to submit data to the regulator to support approval for the new manufacturing process. The initial approval for the biologic included the approval of the process for how the biologic was made. This manufacturing process was called a roller bottle technique, producing the biologic RB in this case. The roller bottle technique is very time and space consuming. The manufacturer wanted to change to a high throughput technique, producing the biologic HT in this case, because using bioreactors improves the yield. However, the high throughput technique may also change the molecule. Therefore, the manufacturer needed to make adjustments to the process to produce the new biologic and demonstrate that the new product was highly similar to the biologic produced by the roller bottle technique. In this new process, the master cell bank needed to be re-established for the biologic HT. In addition to the approval for change in process that the manufacturer was requesting, there was also a need to remove any mammalian-based components in the manufacturing process. This was during the time where there were concerns of bovine spongiform encephalopathy, or mad cow disease. In the manufacturing process change, the manufacturer established two new batch sizes and processes, a 2,000-liter batch for conducting clinical trials and a 15,000-liter batch for commercial processes. These two processes and the previous roller bottle process produced batches that were all analytically comparable with no significant differences observed. The data was part of what was required for the comparability exercise, where the new process needed to demonstrate clinical comparability, analytical methods comparability where methods were to be changed, process comparability, and comparability of the drug product. 3 clinical trials were required by the regulator in order to approve the new process and product. There was a randomized 2-way open-label crossover study to assess the comparability or highly similarity of the pharmacokinetics of the biologic HT and biologic RB with 48 healthy volunteers. The pharmacokinetics and safety were assessed in this study. The pharmacokinetics were found to be comparable. There was also an open-label safety study switching patients from the biologic RB or another comparable biologic to the biologic HT in a different non-cancer indication. Here, 1,127 patients were treated. No new safety signals were detected, and no differences in immunogenicity were detected. Finally, there was a randomized phase 3 equivalence trial in a non-cancer approved indication where 446 patients were randomized to either be switched to the new biologic HT or remain on their current biologic RB. These subjects were treated for 28 weeks. The next slide has the results of this phase 3 clinical trial. In the phase 3 clinical trial, the first primary endpoint was met. Here, the two-sided 95% confidence interval was entirely within the protocol-specified and acceptable equivalence margin. In addition, the second primary endpoint was met. Its two-sided 95% confidence interval was within the protocol-specified equivalence margin. For this study, the chosen equivalence margins were based on criteria usually used to demonstrate bioequivalence in order to demonstrate equivalent efficacy. Overall, the clinical trial demonstrated comparable efficacy between the biologic HT and the biologic RB. In addition, there were no clinically meaningful differences in the safety of the two biologics. The regulator's recommendation was to grant an extension of the marketing authorization to biologic HT for all indications that were given to biologic RB. This included authorization for adult and pediatric patients in non-cancer indications, as well as for adult cancer patients. The data requirements for the approval process is very similar to what a biosimilar manufacturer must provide to prove comparability. What was the biologic in this case study? This biologic was Aranesp or darbepoetin. The process change occurred in the early 2000s and was approved by the EMA in 2008. The process of approval was documented in the Variation Assessment Report for Aranesp. The phase 3 study was a maintenance study where patients on darbepoetin were randomized 1-to-1 to be maintained on the RB biologic or switched to the new HT biologic. This patient population was chronic kidney disease patients on hemodialysis. The open-label safety trial was where chronic kidney disease patients were switched from either the RB biologic or from erythropoietin to the HT biologic. These patients could have been on dialysis or not. This case study demonstrates how extrapolation is used in approving a biologic that underwent a significant manufacturing process change. This is very similar to how a biosimilar manufacturer must approach a submission of its biologic that references an approved biologic. Extrapolation of indications where clinical trials have not been conducted can be justified by the information that is submitted to support extrapolation. Health Canada may authorize indications for biologics and biosimilars that have not been studied in clinical trials. When we examine the regulatory requirements for therapeutic oncology biosimilars, we see that they need more robust clinical data for approval due to a lack of sensitive endpoints. Clinical trials must be conducted in a suitable indication that has a highly sensitive population, for example, active disease, the medication has a large effect on the disease, and the patient population is immunocompetent and not suppressed. It must also be a homogeneous population with a single disease with balanced patient characteristics. Equivalence trials are preferred by regulators, but non-inferiority trials may be permitted with an appropriate rationale. Non-inferiority trials have been used for oncology biologics in conjunction with equivalence trials in non-oncology indications. How are clinical trials for biosimilars designed? The guiding principle is to demonstrate similar efficacy and safety compared to the reference biologic drug, not patient benefit per se, which has already been established by the reference biologic drug. It is for this reason that highly sensitive populations are used, with endpoints where the largest difference is seen when adding the reference biologic to standard treatment. In other words, it would be most obvious in that scenario if the medication was not working as well as it should be. Phase 3 biosimilarity trials are confirmatory trials that evaluate whether the biosimilar and its reference biologic will have the same degree of effect on the disease. If a product has not shown comparability in all of the other characterization of the biosimilar, the Phase 3 trial would not proceed. Please note that the reference for this slide comes from the EMA, however Health Canada uses similar scientific principles as the EMA when looking at clinical trial design but does not have a publicly available document. The next few slides look at how the highly sensitive endpoints used for the 3 oncology biosimilars in clinical trials compares with the initial approval of the biologic. What is the rationale for Bevacizumab biosimilar endpoints? Bevacizumab has been studied and approved in many indications. The following are randomized Phase 3 studies that regulators, like Health Canada and or the EMA, have used for the approval of Bevacizumab Avastin. These are all trials in the first-line setting so that patients were not immunocompromised by previous treatments. All 3 of the studies compared the addition of Bevacizumab to the standard of care chemotherapy, irinotecan-based therapy for metastatic colorectal cancer, and paclitaxel plus carboplatin in non-small cell lung cancer and ovarian cancer. Note that first-line ovarian cancer is not a Health Canada approved indication. Progression-free survival was statistically better across the 3 studies, but numerically not as large of a change. Same applies for overall survival, with ovarian cancer not being statistically significant. The objective response rate in lung cancer was both statistically significant and numerically different. Of the 3 disease states, this was the endpoint that would be most sensitive to detect differences between the biologic and the biosimilar. For the approval of a biosimilar Bevacizumab, objective response rate was used as the primary endpoint. Both MVASI and Zirabev demonstrated equivalence to the reference Avastin. Also included is the initial clinical trial in a non-small cell lung cancer where approval of the biologic was granted. Also note that the secondary endpoints from the trials were highly similar. What is the rationale for Trastuzumab biosimilar endpoints in metastatic breast cancer? The pivotal study that resulted in the approval of Trastuzumab for metastatic breast cancer had a 90% power to detect a 50% increase in the median time to progression and required 450 patients to detect that difference. The study randomized 469 patients to receive either chemotherapy alone or concurrently with Trastuzumab. In this study, 188 of the 469 patients were treated with paclitaxel as their chemotherapy. The rest were treated with anthracyclines. As a result of the cardiac toxicity observed in this study, Trastuzumab is not routinely given concurrently with anthracyclines. Therefore, the patients treated with paclitaxel are the only population that is reflective of the current treatments, and it was the data of 188 patients that served to approve the original biologic. In this study, the patients treated with paclitaxel before their chemotherapy demonstrated that the primary endpoint, time to progression, was significantly better, but was very short. Overall survival was also not significantly better. The objective response rate in this study was statistically significant, and the numerical difference was large enough to detect any differences between the biologic and biosimilar. As a result, this was used in the patient population for clinical trials to demonstrate equivalence. For the approval of a biosimilar Trastuzumab, we can see that in metastatic breast cancer, the equivalence of the biosimilars to the reference biologic. The objective response rate is equivalent, and while numerically higher than historical trials, it remains equivalent between the reference biologic and the biosimilars. Note that the secondary endpoints were also similar. The rationale for the Trastuzumab biosimilar endpoints in neoadjuvant breast cancer is different than that of metastatic breast cancer. The clinical trial that resulted in the approval of the reference biologic in the EU was the NOAA trial. This consisted of 235 patients randomized to chemotherapy plus or minus Trastuzumab prior to surgery. The primary endpoint of event-free survival was statistically better with the addition of Trastuzumab, but the difference was not as large as pathological complete response rate, which was statistically significantly different and numerically greater. Pathologic complete response rate is a more sensitive endpoint than event-free survival. Note that the neoadjuvant use of Trastuzumab is not a Health Canada or FDA approved indication. For early breast cancer, the pathological complete response rates are equivalent and similar to the historical results that are still indicative of current treatment. Note that in the filings to the EMA, it was noted that in the Kanjinti and Untitled trials that the pathological complete response rate was lower with the reference biologic than expected. This was accounted for in the EMA's approval of the biosimilar. As well, note that Trastuzumab had a non-inferiority study that was conducted for non-medical reasons, as there was a manufacturing process change that the regulator requested a confirmatory clinical trial to look at efficacy and safety. Trastuzumab was already demonstrated to be equivalent in the metastatic setting. In approving new drugs for cancer, regulators prefer endpoints like disease-free, progression-free, and overall survival. However, the regulators acknowledge that these endpoints may not be feasible or sufficiently sensitive for establishing the similarity between biosimilar monoclonal antibody and a reference monoclonal antibody. Shorter-term efficacy endpoints, such as tumor response or objective response rate, could be acceptable for initial marketing approval. In looking at adjuvant breast cancer, we need to put the endpoints in perspective. A recently published joint analysis of the NSABP B31 and NCCTG N9831 studies demonstrated that the 10-year overall survival was increased by adding Trastuzumab to the chemotherapy solely in the adjuvant, not neoadjuvant setting from 75.2% to 84%. The size of the clinical trials ensured that the difference was statistically significant overall, as well as in the various subgroup analyses that were planned. However, the difference in overall survival when comparing patients that did not receive a year of Trastuzumab to those that did showed an 8.8 absolute difference at 10 years. Over the coming years, the biosimilar manufacturers will be reporting the survival endpoints for their clinical trials as more long-term data emerges. The rationale for Rituximab biosimilar endpoints follows similar reasons for the previous two biologics. Rituximab is used in several oncology and non-oncology indications. 3 sensitive diseases can be used in the clinical trials for Rituximab approval: follicular lymphoma, diffuse large B-cell lymphoma, and rheumatoid arthritis. In treating lymphoma with standard of care chemotherapy, overall response rate is the most sensitive endpoint to demonstrate if there are any differences in the activity of the biologics. When comparing the two lymphoma indications, overall response rate is more sensitive when studied in follicular lymphoma, and Rituximab is added to CVP chemotherapy. It is also sensitive in diffuse large B-cell lymphoma when Rituximab is added to CHOP chemotherapy. However, there is a smaller absolute difference. The same applies to CLL. Most biosimilar clinical trials have been in follicular lymphoma when studied for the oncology indications. The two biosimilars that have been approved in the EU use studies with patients who had follicular lymphoma treated with CVP chemotherapy as the basis for their approval. The two biosimilars were also studied in rheumatoid arthritis. If their lymphoma trial was conducted in an equivalence design, then the rheumatoid arthritis study was in a non-inferiority design, and vice versa. In follicular lymphoma, the overall response rate was highly similar between the reference biologic and the biosimilar. Overall survival was also measured, and at 2 years was similar as well. A biosimilar candidate that has not yet been approved by a regulator was studied in diffuse large B-cell lymphoma. It demonstrated non-inferiority in a trial with patients treated with CHOP chemotherapy. A determination of biosimilarity has not yet been announced. In conclusion, endpoints used to approve a biosimilar are highly sensitive to detect that there are no differences from the reference biologics effects on the disease studied. The endpoints for biosimilar clinical trials are carefully selected for each individual indication. Authorization of indications for biosimilars by Health Canada in diseases not studied by clinical trials is based on good scientific rationale. Extrapolation has been used by biologic manufacturers even after biologics have been approved by a regulator. The Pan-Canadian Oncology Biosimilars Initiative is a joint initiative of the Pan-Canadian Pharmaceutical Alliance and managed by Cancer Care Ontario on behalf of all provincial cancer agencies. The PCPA is an alliance of 13 Canadian provinces and territories, including 3 federal drug plans that collaborate to negotiate for brand name and generic drugs to achieve greater value for publicly funded drug programs and patients.